How CRISPR can fix our genes, the future of genetic engineering. Have you ever imagined that one day we could edit our genes to fix genetic mistakes? A new tool called CRISPR-Cas9 makes this possible. Today we'll learn what CRISPR is, how it works, and how it's used to treat genetic diseases. What is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It was first discovered in bacteria, which use it as a defense system to fight viruses. Scientists realized they could use this same system as a kind of molecular scissors to cut DNA at specific places. How does it work? Cas9 enzyme acts like scissors to cut DNA. Guide RNA gRNA acts like a map, showing Cas9 exactly where to cut. When Cas9 cuts the DNA, the cell tries to repair the break. Scientists can use this repair process to insert a correct version of a gene or remove a harmful one. Scientists have used CRISPR to treat genetic diseases such as sickle cell anemia and cystic fibrosis, create cancer therapies, improve crops to make them more resistant to pests and disease. Challenges and ethics. Even though CRISPR is powerful, it raises some important ethical questions. Should we edit genes in human embryos? What if we make mistakes or cause side effects? Scientists are still trying to balance innovation with responsibility. Conclusion. CRISPR is changing the future of medicine and biology. It gives us the power to fix DNA, but also the responsibility to use it wisely. CRISPR equals the tool that lets science rewrite life's code.